After doing many hours of research, I've finally done it. I found it. I found the most amazingest, craziest thing. Gene therapy! Hey, I'm Suresh, and today we're going to be talking about, yep, that's right, you guessed it, gene therapy. Okay, let's take a step back. What is gene therapy exactly? It's basically when you take viral vector and you insert a specific gene into a cell. Okay, there were a few words that you probably didn't understand, but I'll break it down even further. Viral vectors are these viruses that have been stripped of their really bad virus DNA, and then like a good gene inserted inside of them. After that, that gene is inserted into the body, or rather, the virus is inserted into the body and delivers the gene. It does this because it's this special kind of virus called a retrovirus. It's pretty nifty, trust me. So in essence, they're good viruses. What they do is that they remove almost all of the DNA and RNA from the virus, depending on what type of virus it is. And then they replace it with new genes that are specific to a certain disease. So let's say that you're using a viral vector to insert a gene for sickle cell anemia. Then you'd insert the gene that you're looking for in sickle cell anemia. It really just depends. Viral vectors are typically made of these viruses called retroviruses. What's special about them is that they actually insert their own RNA and DNA inside of the cell. That's pretty cool, because most viruses don't actually do that. When dealing with viral vectors, there are two main types that we're going to be talking about today. These are the adenovirus and the adeno-associated virus. These are actually the two most prevalent and used viruses in the gene therapy field today. So both of these have their pros and cons. It's not just like a one-way street. Adenoviruses are used when you want to insert a very long gene. A gene that has lots of information, and you need to get it in there quickly, like really quickly. The problem with adenoviruses is that they have like a sort of flu-like effect that lasts for a few hours after it's injected. Another problem with it is that it really incites like a huge immune response. So what that means is that when it's inserted into the body, much like any other virus, the immune system goes and hunts it down, which reduces the effectiveness of the virus. Adeno-associated viruses, on the other hand, are different in the sense that they have a smaller packaging rate, one, and two, they last in the blood for a bit longer. They take a bit more time to incite like a gene response, so it takes a bit more time, and the chance of a protein expression is significantly lower. The reason that people are using adeno-associated viruses, though, more than adenoviruses, is because adeno-associated viruses, one, they don't have any known effects to the human body, and two, they don't incite much of an immune response, which is pretty freaking great, because you don't want to get sick every single time you get an injection, right? The usage rates for gene therapy are pretty freaking crazy. Like, you can use it for delivering CRISPR, or you could even use it for delivering a certain gene. For example, P53. I'm going to be talking about the latter right now. P53 is basically just this like tumor suppressor cell. It stops tumors in its tracks, and it makes sure that the cell doesn't continue to mutate and perform mitosis if it's mutated and there's genetic distortion in its genome. What people are doing with P53 is that they're taking P53 and inserting it into the adenovirus. Fun fact, the first gene therapy drug that was actually approved was called Gendicine. Gendicine was this ADP53 drug that was approved in China. It was super effective, or at least that's what they said. But when they took it to America and they labeled it as Advexin, supposedly it didn't make it through, as it didn't have substantial amount of evidence. I mean, hypothetically this would work. You're basically just inserting the main gene or protein that's necessary for tumor suppression to work, which is actually mutated in more than 50% of all cancers, and you're inserting it back into our bodies so the cell cycle can work again. That sounds really awesome, like initially, because it makes sense. By inserting this gene, you can essentially cure 50% of all human cancers. But this isn't what happened. When they took it to America and they called it Advexin in 2008, it didn't make it through the FDA pipeline approval process because it didn't have enough substantial evidence. I don't know why this is. I'm gonna look more into this, but it's really super intriguing. Right now, there are about 12 or so gene therapy drugs in the market, and it's a huge industry. I think there's a lot of potential for gene therapy in the future, and I think that's where we're going in the future, especially with CRISPR, because right now, viral vectors are one of the best ways to deliver CRISPR. Okay, that was it. Gonna see you in the next video. Peace.